Hello everyone, welcome to our webinar about our ingredient Sibelius Chamomile. My name is Melina Saoulidou and I'm the marketing executive at Sibelius. And with me today is Dr. Kieron, the Chief Scientific Officer at Sibelius.
Hi there.
So today we're going to talk about our Sibelius Chamomile and the science behind that. So just a short introduction about us. We are an R&D company. We're developing natural and clinically researched ingredients. All our ingredients are developed with a great focus on traceability and quality control. And that's all around our ChronoScreen platform, which help us to identify and launch only the best ingredient from the best source. Meeting consumer demand is one of our top priorities. After the outbreak of COVID-19 pandemic, people are getting more conscious about their health and they are looking for natural alternatives. So here at Sibelius, we make sure that our ingredients support a range of consumer demands, including sleep, relaxation, skin, digestion, memory. Next slide, please. So let me introduce you Sibelius Chamomile. Sibelius Chamomile is a branded, traceable and scientifically evidenced high-quality Roman chamomile extract, which is specially developed to support inflammation, sleep, digestion, skin, allergies and mood. Currently is available in tincture and the powder is coming soon. So Sibelius Chamomile is one of our best-selling ingredients. Consumers love it. We have received great feedback on that. So talking about traceability, traceability is one of our top priorities here at Sibelius. We have an exclusive UK source with the largest grower of Roman chamomile in European region. Our proprietary source is able to ensure the consistency of high quality ingredients that is propagated and rain-fed, promoting the sustainability of our process. So anti-inflammatory, antioxidant, skin health and digestive health are some of the healing applications of chamomile that are well documented. So the German chamomile and the Roman chamomile are the most two common species of chamomile. So even though they have similar look, they have significant differences in their biochemistry. So now I'm going to hand it over to Kieron to discuss about the supporting science around Sibelius chamomile.
Thank you, Melina. Thank you very much for the introduction there. So yeah, I'll talk to you a little bit about Sibelius chamomile and how we've developed that into the ingredient that it is, taking you through the various stages of development that we do at Sibelius, all the way through from our ChronoScreen approach through to trying to put some scientific evidence behind it. So as with all our ingredients, we're very particular about the source we go to. And obviously, Melina, thank you very much for introducing the source for chamomile there. You know, there's a lot of considerations going into it about the sustainability of the source, the quality of the source. And we use our ChronoScreen approach to take a very careful selection of that source. And you can see with our chamomile, we get a nice extension of lifespan on our ChronoScreen platform there. This shows we're having really good beneficial effects on cellular health in our model organism C. elegans. And this is our, for anyone who hasn't heard about ChronoScreen before, this is essentially our screen that helps us to differentiate between different sources of extract, looking for ones which have the most anti-aging, the strongest anti-aging effects on a model system. So obviously, we want to look at what that extract is doing. We've picked a nice source, which is very sustainable, and we've got good activity and biological assay coming from that. But how does it translate through to benefits in people? As Melina already mentioned earlier, we know that chamomile has a long history of use and benefits in terms of anti-inflammatory effects. And we've done some preclinical work looking at anti-inflammatory effects for our chamomile extract. And in particular, we see some really nice, strong anti-inflammatory effects coming from that when we treat human cells with the chamomile extract. So for example, you can see it at the top here, if we look at different cell lines, so either an adipocyte cell line or a neuroblastoma cell line, if we compare our control condition in the bright orange through to our treated next to that with the chamomile, you can see we get significant knockdowns in the basal conditions from the treatment with the chamomile. To the right of those as well, if we try and induce inflammation in those cell lines by using a pro-inflammatory cytokine such as IL-1 beta, which is really trying to make pro-inflammatory conditions in these cells, we can actually attenuate the increase. So what we're looking at in these charts at the top there is an increase in the release of TNF alpha, which is an inflammatory cytokine and would indicate higher levels of inflammation in those cells. So this work has been published in the Journal of Ethnopharmacology, if people are interested to go and look at that. We've also see effects on multiple other markers in these cell lines, so please do go and look at that study if you want to find out more on that. In addition to that, we've tested some other cell lines in data we haven't published yet. So for example, we get nice anti-inflammatory and antioxidant effects in endothelial cells, so the cells that line our vasculature. And you can see that on the plot down at the bottom there, where we look at inducing the release of reactive oxygen species through hydrogen peroxide treatment in the endothelial cells, and you can see that we get a dose-dependent reduction in the release of in the release of reactive oxygen species in the presence of chamomile. So we see these nice anti-inflammatory and antioxidant effects in some in vitro systems. We've taken that further as well and used the Biomap Diversity Plus panel assay to analyze this. So this essentially is where we're treating a whole suite of different cellular models with our chamomile treatment and looking at the response of multiple different biomarkers on there, so we can get an idea of anti-inflammatory and other effects that are occurring on it. So that's summarized in the plot on the right here, where we've got these different cellular models represented across there, and with the colored traces going from a low dose in green through to a high dose in red, we can see different responses to the treatment with the chamomile. And as you can see, we get some really nice strong effects in several of these panels here, where we're reducing the levels of multiple inflammation markers. So for instance, in this BT panel in the middle there, you can see, which is a model of T-cell dependent B-cell activation, we get strong reduction in things like IL-6 and IL-17. So pro-inflammatory markers, again, being knocked down in that model. Similarly, we can see in this panel, the HDF3-CGF panel, which is more a model of tissue remodeling and wound healing. Again, we get knockdown of multiple inflammatory markers in there, which ties in with what we know about chamomile being good for skin health, for instance. I'll also sort of point out that if you look at the three panels on the left-hand side, the 3C, 4H, and LPS, these are all panels that have endothelial cells, just like we looked at on the previous slide. And you can see we get a knockdown of vascular cell adhesion molecule 1, VCAM1, which is again an inflammation marker in the vasculature. So we're getting some really nice anti-inflammatory effects coming from our chamomile when we treat human cells with them, and particularly across different areas, in fact, coming through there. Now, we obviously want to understand certain mechanisms of actions that might underlie these anti-inflammatory and immunomodulatory effects that we're seeing in the in vitro data. So part of what we're able to do with the data we've developed, so particularly with the Biomap, for instance, we can start comparing that data against databases of known treatments that have been run on that same platform, and ask the question: do we show potential functional similarity to those treatments? And what we found very interestingly for the chamomile actually is we get nice strong similarity to COX-2 inhibitors. So COX-2 inhibitors are the target that non-steroidal anti-inflammatory drugs are targeting there. And what COX-2 is doing, cyclooxygenase 2, is metabolizing arachidonic acid into prostaglandins, which are these pro-inflammatory signaling molecules. So the COX-2 enzyme gets induced when you get local inflammation occurring there, and that local sort of damage and signals coming through to induce local inflammation. And what we're actually doing is inhibiting the activity of that enzyme with the chamomile. much in the same way that non-steroidal anti-inflammatory drugs would do. So we've got that same activity which would contribute towards this. It's also worth noting that prostaglandins play other roles in the body related to sort of the smooth muscle contraction, GI tract, platelets. There's other roles for these prostaglandins in the body, and in particular, we don't want to actually inhibit COX-1 activity much. And it turns out that the chamomile appears to be a COX-2 specific inhibitor, which is very nice for us to see that. And it may be that some of these activities related to prostaglandins are actually playing through into what's been shown previously for Roman chamomile, in that it has smooth muscle relaxant properties. So when we're talking about smooth muscles, it's not your skeletal muscle where we're contracting, moving our body. These are the muscles found in places like the gastrointestinal tract, the cardiovascular system, and they contract without voluntary control there. And the chamomile has the ability to have a relaxant effect on these muscles, which you can see, again, playing through to potential benefits that have classically been linked to chamomile, such around digestive health. So you can, stomach cramps, these kind of things, helping to ease those sort of burdens. So we see some nice beneficial effects on chamomile coming in these anti-inflammatory, immunomodulatory areas. And actually, one of the first interesting areas of interest we had for chamomile was related to hay fever. We thought there may be allergic rhinitis. There may be some benefits to the chamomile in this area. So we did a small observational study a few years ago where people taking the tincture, and 71% of the participants involved in this actually reported their improvements to their hay fever symptoms. What was interesting as well, though, was actually several of the participants also noticed improvements to their sleep, as well as improvements to their digestive comfort. So these were improvements they're reporting completely independently. They weren't asked specifically about here. So ties in very nicely with as we've talked about, traditional applications of chamomile, and we're seeing those effects represented by our chamomile in studies when we weren't even looking specifically at those. So obviously we're interested to take that further forward. You can see we've went on and we've done a similar observational study on sleep in 18 healthy volunteers, and we could see that we got really nice improvements in people's sleep. So for instance, you're looking at here the number of wake-up events over 4 weeks, from a baseline number of wake-up events through to 4 weeks of having taken the chamomile tincture, and we get a sort of a 90% reduction in the wake-up events occurring. We also saw like a 7% increase in sleep duration and also improvements in self-rated sleep quality coming through this period. So it really kind of ties in. This chamomile does seem to be improving people's sleep, and it's an area of further research for us. So yeah, I'll obviously please do go and look at the papers that were mentioned there, and keep an eye out for further publications we'll make in this area.
That's great, Kieron. Thank you very much for the information. I would like to ask some questions before we move forward, if you don't mind.
Sure, please.
So what do we think are the main active compounds present in the Roman chamomile extract?
Sure, that's a very good question. When people think about chamomile, classically, you tend to think of compounds like chamazoline. There's also angelic and tigalic acid tend to be linked to the activities in there, as are flavonoids such as apigenin, very much classically linked to biological activities in chamomile. And there's every chance that these compounds are very important in our extract as well. What I'd also say on top of that, though, however, is from the work we did with Biomap, we saw some interesting connections to coffee extracts coming through from the activities there. So there's a very strong chance we're looking at other phenolic compounds such as phenolic acids, caffeic acid, these type of compounds present, similar to what's present in coffee, which were known to have anti-inflammatory effects as well, could be playing an important role in our extract too.
Okay, great. Thank you very much. So, and the second question I have is that, given what we know about Sibelius chamomile so far, where do we think it has the best application to support our health?
Yes, yeah, another good question there. I suppose, you know, obviously we're seeing some really promising data in the area of sleep and obviously the relaxation linked to that. And chamomile's obviously got historical uses as anxiolytic. So I think that's definitely one area where we will look to demonstrate the benefits of the chamomile. In addition to that, I think the anti-inflammatory effects and the immunomodulatory effects that we're seeing from our preclinical work will actually support potentially some very strong applications in other areas. I think, you know, digestive health is one area where we're really going to see potential benefits, both from in terms of those anti-inflammatory effects, but also, as we touched on, the smooth muscle relaxant properties. And similarly, these benefits may be very useful in women's health in terms of things like menstrual cramps, etc. Having the smooth muscle relaxant activities around the uterus, which underlie menstrual cramps, for instance. So there's a number of areas where I think the combination of activities we're seeing from the extract could be very interesting.
Perfect. Thank you very much. So moving forward to the features, as mentioned earlier, Sibelius chamomile is derived from a traceable source. The tincture is standardized for total phenolic content of minimum 0.05%. Regarding its flavor, its herbal characteristic flavor can be easily masked, and our in-house team can support you with your NPD process. And lastly, in terms of stability data, it has long shelf life, 3 years. Moving forward, regarding the delivery formats, Sibelius chamomile can be incorporated in a wide range of formats, such as capsule and tablet, oral nasal sprays, tincture, food, lotions, and drinks. Thank you very much for watching. Kieron, thank you very much for the great information and for helping us to understand the science around chamomile.
Thank you.
Please do not hesitate to request a free sample and download the white paper to learn everything about chamomile. And if you have any questions, please get in touch. Thank you.